Namaste. Namaste. Myself Name-Shivi Saxena, Faculty of Physiotherapy and Diagnostics, and welcome to Digital Version 2.0. So, my today topic is Indole production test. So, firstly, introduction of Indole production test. This test demonstrates the ability of certain bacteria to decompose the amino acid tryptophan to indole, which accumulates in the medium. Indole protection test is important in the identification of bacteria. So most strains of E. coli and other species break down the amino acid tryptophan with the release of indole. This is performed by a chain of a number of different intracellular enzymes. A system is generally referred to as tryptophase. It is used as part of the IMBIC procedure, uh, a test designed to among a member of the family Enterobacteriaceae. A variation on this test using enriched reagent that means using ethyl alcohol in place of isoamyl alcohol and developed by Paul English is used when performing the test of fermenters and anaerobes. So, principle of indole test. Firstly, tryptophan is an amino acid that can undergo determination and hydrolysis by bacteria that express tryptophase enzyme. Indole is generated by uh, reductive uh, dimension from tryptophan via the intermediate molecule and indole pyruvic acid and tryptophanase catalyze the dimension reaction during which the enzyme, that means NH2 group of the tryptophan molecule to remove. Final products of the reaction are indole, pyruvic acid, and ammonium and energy. Pyridoxal phosphate is required as a coenzyme. Okay, so when indole is combined with Kovac reagent, that means which contain hydrochloric acid and dimethyl aminobenzaldehyde in ml alcohol, the solution turned from yellow to cherry red because amyl alcohol is not water soluble. The red coloration will form in an oily layer at the top of the broth. Okay, in the spot test, so indole combine in the filter paper matrix at an acid pH with p dimethyl amino cynamaldehyde, that means DMACA, to produce a blue to blue-green compound. Indole spot reagent has been reported to be useful in detecting indole production by members of the family Enterobacteriaceae and certain anaerobic species. So, reagent used in indole test. Firstly, ingredients per liter. Okay, so indole spot reagents. First, p-dimethyl amino cyanomaldehyde, that means DMACA, 10 gram, and hydrochloric acid 37%, 100 ml, and deionized water 900 ml. Okay, and Kovac reagent. So, uh, second reagent may indole Kovac reagents. So, P dimethyl methyl aminobenzyaldehyde 50 gram and hydrochloric acid 37% to 50 ml, and amyl alcohol 750 ml. Okay, and adjusted or supplemented as required to meet performance criteria. Okay, so next step is procedure of an Indole test. So first step, take a sliced test tubes containing 4 ml of tryptophan broth. Okay, and second step, inoculate the tube uh, aseptically by taking the growth from 18 to 24 hours culture. Okay, and third step, incubate the tube at 37 degrees Celsius for 24 to 48 hours, and add 0.5 ml of Kovac reagent to the broth culture. And last step, observe for the presence or absence of drain. Okay, Indole second method, Indole spot reagent DMACA procedure. So first step is place several drop of the Indole spot reagent on a piece of filter paper. Okay, and with the inoculating loop of wooden applicator stick, pick a portion of the 18 to 24 hours isolated colony from a non-selective media and rub it all onto the reagent saturated area of the filter paper. Okay, and last step is examine immediately. Okay, so result interpretation of Indole test. Positive result seen in formation of a pink to red color, pink to red color in a reagent. So pink to red color. So this picture is seen, and in the reagent layer on top of the medium within second of adding the reagent, or and example, E. coli and Haemophilus influenzae, Klebsiella oxytoca, Proteus species, not Proteus mirabilis and Proteus penguin. Okay, and Anthrococcus bacillus and Vibrio species. Okay, and etc. And negative result is shown no color change even after the addition of appropriate reagent. So this image show that this image wait so that this image negative Indole test, this form no change color. Okay, and examples, so Actinobacillus species and most Bacillus species, Neisseria species, and Proteus mirabilis and most Haemophilus species, Lactobacillus species, Enterobacter species, and Salmonella species, Pseudomonas species, and it is in. So Indole spot reagent results, so positive reaction. So show the development of a blue color within within 3 minutes, and negative reaction, the development of a pink color within 3 minutes. Okay, so spot Indole test procedure. So method first, place several drop of 1% p dimethyl amino senomaldehyde reagent on a piece of filter paper until saturation, and with an inoculating loop of wooden application stick, pick a portion of the 18 to 24 hours isolated colony from a non-selective media and rub into saturated area of the filter paper, and observe the color development within 1 to 3 minutes. Okay, so expected results positive. A positive reaction is denoted by the appearance of a blue to blue green color change on the bacterial smell within 2 to 3 minutes, and negative result shows negative reaction remain colorless or light pink. So note down the note down Positive reaction is red violet in the case of Providencia alkylphenase bacteria. Okay, so uses of indole test. First use to differentiate protease milliabilis indole negative from all other protease species, that means indole positive. And second use to differentiate Klebsiella pneumoniae, that indole negative, from Klebsiella oxytoca indole positive. And third, to differentiate Citrobacter tremula indole negative from Citrobacter cocci indole positive. And quality control for indole test. Positive control use E. coli NCTC 10418 and negative control protease milliabilis NCTC 10975. Okay, so limitations of indole test. Indole test may be used as an aid in the identification and differentiation of gram-positive and gram-negative organisms. Additional biochemical tests using pure cultures is recommended for complete identification. The tube test is more sensitive method for detection indole than the spot test. And when performing a spot test, Kovac indole reagent may used as a substitute for the spot test reagent. However, Kovac indole reagent, when used as a spot test reagent, is less sensitive in detecting indole than the indole spot reagents. And Kovac indole reagent is not recommended for use with anaerobic bacteria. The indole spot reagent is suitable for anaerobic use. Okay, and since peptones have been shown to vary with regard to their suitability for use with indole testing, and media selected for indole determination should be tested with known positive and negative organisms to ensure suitability. And next, media containing glucose should not be used for indole testing uh, due to the formation of acid and products which have been shown to reduce indole protection. Muller-Hilton agar should also not be used for this test because tryptophan is destroyed during acid hydrolysis of gas. Okay, so media containing dyes such as McConkey's and EMB are unsuitable sources of inoculum due to possible carryover of dye and subsequent interference of indole color interpretation. And next, limitation, indole positive colonies have been reported the cause adjustants indole negative colonies to appear false positive due to diffusion of indole into the medium. To avoid false positives, select colony of different morphologies that are separated by at least 5 mm for indole testing. Okay, so this session is completed, and any query in mention comments box, and thank you so much. Namaste.